These slides. So it's a true pleasure to be here in this panel. Just like everybody else, I would like to point out that this is a big science project. In another parallel life that I live, I study science. By scientific means, we render large-scale maps of science and try to help people understand what areas of science are evolving, how quickly, how life trajectories and career trajectories unfold over the landscape of science, and also how to make teams productive across disciplinary and also institutional boundaries. So this project is slightly different, but it's still involving maps. Here we are interested to create a map of the human body at the single cell level, so trillions of cells. And how do you go about this, given that we are all very, very different? We have different sexes, different ages, different ethnicities. We have different BMIs, different ways of living our lives. And it's non-trivial to actually create one single map. And maybe we end up with a bunch of maps at the end. So in this initial set of 2 years, which we have been running for inside of Hubmap, we have been working very closely with some of the tissue mapping centers, especially those focusing on spleen and kidney. And we have also interacted very closely with all the Hive component teams. And it was great to see their presentations just now. So some of you might wonder, what in the world is a common coordinate framework, or CCF? And there are now even comics about this. And ultimately where you want to go is to that npm install Hubmap Atlas. But there's a lot of confusion in the middle of how we actually get there from the data we have so far, but also data that other consortia are now contributing. And I think a major point that I think all collaborators here try to make is that we can't do it alone. I think we need to share data, share code, and share functionality, and ultimately also education training materials with all of you and all of you who want to collaborate. Ultimately, it is a matter of taking a common coordinate framework that consists of ontologies and reference objects. And I think Nick and others already mentioned ontologies and how important they are. They are the existing understanding of the structure of anatomy, but also of existing cell types. For instance, the cell ontology, a hierarchical structure that is used to bring order to the different cell types, even though there's not necessarily agreement on cell type names, but also ultimately on biomarkers, which could be quite diverse, as you already have seen in Sif's and also Niels's presentations, and also Mike's and Coltrop-Nell's presentations. So here you use that CCF to ultimately enable biomedical experts to semantically annotate tissue samples and to precisely describe the location of these tissue samples in the human body. And again, every single body is slightly different, but we call that process registration. To then also use the common coordinate system to align multimodal tissue data extracted from different individuals to a reference organ or coordinate system. That's very similar to what Alan, for instance, does for the brain. We are interested to create consensus organs into which you can register new tissue samples. And in order to arrive at these consensus organs, we have to understand what is normal and what different types of normal do exist. And ultimately, we want to provide tools for searching and browsing Hubmap data in the portal, but also via APIs and via other portals at multiple levels, from the whole body down to the single cell level. And we call that exploration. And we are using here, of course, keywords of metadata, but also ontology terms and spatial locations for browsing. To make that even feasible, we have a very initially simple understanding of the human body. We have a nested structure of cells being part of certain functional tissue units, being part of larger units in the human body, being segments or tissue blocks that are cut out ultimately of organs. And then of course the organs need to be registered into the human body. Now this by itself already is non-trivial because different systems use quite different reference systems for these different levels. And even organs that different organ societies are using, they might use very different organ reference systems. But ultimately, we want one human body, maybe two for different sexes, into which you then register in the organs, but also the tissue pieces, etc. Ultimately, we also want these anatomical structures and cell types to be spatially explicit. So therefore, we have the registration, but we will also want them to be semantically explicit. So we want them to use terminologies that pathologists, anatomists, clinical doctors agree upon. And that's another major challenge within Hubmap. In order to make that all work, we are using so-called AST tables that some of you might be now familiar with, which stands for anatomical structures and cell types, which ultimately connect different cell types to anatomical structures and substructures, but also connect further down to subsets of marker genes and protein markers and other metabolites and lipids and many other assay types that you already saw in the previous presentations. So ultimately, there are quite a number of cells. We have about 600 unique ones identified right now across the different organs that Hubmap cares about, and also quite a number of anatomical structures. And there is limited agreement of how to name all these things. And this continues also on the marker genes. So just agreeing on nomenclature for those elements is non-trivial. Here you see an example of the kidney AST table, which was published by KPMP. So we actually are very grateful for them to invent these tables. And in the meantime, we have now created tables for multiple organs. We have monthly meetings which bring together organ experts from different organ societies. And again, there are pathologists and anatomists and clinicians and biomolecular experts. And again, the language it's very much like a Tower of Babel. But what we do together is to review and approve these existing tables, to formalize and unify the table design language, and also to discuss the table usage. And currently, we are working on converting these manually created tables into more machine-readable formats. We compare also these tables with the terminology used in Uberon and cell ontology and other gene and protein ontologies. And then, of course, we identify what is actually in the Hubmap data, what anatomical structures are covered in there, what cell types are discovered in that data, and what biomarkers are currently used in Hubmap and associated with certain cell types. And we try to get to a good match between the tables and the ontologies and what comes out of the data. And it matches partially right now. It doesn't fully match, as you can imagine. So if you're an expert and you're interested to join this effort, let me know, let me know, because I don't think we have the link here active. Now, in these table meetings, they were actually inspired by a recent NIH HCA meeting that happened in March, which Peter Hunter from SPARC, James Gee from BICCN, and myself had the pleasure to organize a CCF session there. And the idea came up that we would all much benefit from actually having agreement on anatomical structures, cell types, and biomarkers. And so in the meantime, we have managed to also invite and engage major anatomists and ontologists, including also Chris Mongel and Mark Moosen and others that some of you might be working with. We have a Slack channel that you are very welcome to join. And this also helps organize the discussion surrounding different organs, which are quite different from each other. As you can imagine, that the lung is quite different from the heart, is quite different from the kidney, all have different affordances and inspire different innovation. And still, some innovation developed for one organ might as well inspire other advantages and advancements for other organs. Ultimately, the yellow marked organs are the ones we have been focusing on first. The column headers here are different consortia, partially funded by NIH, but also funded otherwise. And you get to see how many consortia care about what organs. So it's quite a variety of efforts that are focusing on the same organ, but are some are also pretty unique to one or two efforts. In the meantime, we also have started to identify vasculature tables. We are very, very interested to explore using the vasculature as a unique addressing system for the human body, because every single cell, almost every single cell in the human body needs to be fed by blood or will die very quickly. And so this then can be represented for the kidney in this table up here. And then a flattened version of that table that is much more machine readable is shown below, just so that you get an understanding of how these flattened tables look like currently. Ultimately, these tables work across multiple scales, from the organ scale to the functional tissue unit scale, down to the single cell micro scale. And again, you can unfold this again and again. And of course, ultimately, every one of us will have our kidney glomeruli in different places, but I think about 1 million is in each one of our kidneys. So some of this placement is stochastic, but other parts are potentially very well defined. Like for instance, functional tissue units might have a very well specified spatial structure, and we are trying to understand more about the spatial structure of these functional tissue units using cell segmentations, but also ultimately using these tables to understand what number and type of cells is typically in functional tissue units across organs. Ultimately, the usage of the tables works as follows. So we take tables and we use them to then crosswalk these terms over to existing ontologies, identifying missing terms or different named terms so that we can agree on nomenclature as needed. We are also walking them over to the cell types ontology. Niels just beautifully demonstrated the tissue or the RUT browser, which he developed with Rahul Sahid. So here you have a tree again. So the anatomical structure is currently a autonomy, a tree. Similarly, the cell types are currently a tree. And ultimately also we use this information in the AST tables to create reference objects. And currently we are using the data from the Visible Human project. Ultimately also going down to the functional tissue unit, which comes from the data, the Hubmap data. In general, the process is as follows. The anatomical structure tables here again used to get to autonomies, but also knowledge graphs, which are needed for interlinking Hubmap data to the much richer ontology data that exists out there, which ultimately also links to diseases, and also taking the AST+B tables to create an object library. Then there is a so-called registration user interface, which we use to spatially and semantically register tissue blocks in reference organs. And then that data gets stored in the IEC data store, which Nick presented. And then an exploration user interface currently separate from the portal is used to spatially and semantically browse tissue data. In year 3, we will integrate this CCF search and browsing based on ontology terms and based on spatial location into the portal. And then you can use this kind of functionality also directly in the portal. Interestingly here, we are using collision data from tissue blocks colliding with 3D structures in the reference organs to do semantic annotation automatically. And as you will see, users can, of course, delete tags that were automatically generated. They can also add tags. But I think it's not all up to them to add all these many, many tags, which might be too time consuming on their end. Just to give you a more understanding of the ontology behind all of this, there is, of course, a provenance ontology. We need to know the age and sex and ethnicity and BMI, etc., of the donors. This makes a huge difference in terms of what we ultimately can do with that data for the CCF construction. There's also a semantic ontology that again assigns anatomical structures, cell types, and biomarkers to tissue blocks and tissue sections. And then there is the quite unique here spatial ontology, which really keeps track of the size of tissue blocks, their orientation in 3D, but also their positioning in 3D in reference to an organ. And so again, here is this nested view cells inside of functional tissue units, inside of organs, inside of a human body, inside of the overall Hubmap reference system. And there is a paper that is in preparation, which will, if everything goes well, come out with the portal release. So if you want more details, it will be in there. We have also made available the reference organs, which might be interesting to some of you who are interested to explore tissue registration. So feel free to check that out on our GitHub website. So coming over to registration, some of you might keep track of the spatial position of tissue blocks via photographs or even videos of tissue extraction sites across the different TMCs and other tissue generating centers that we have in Hubmap. This is actually quite diverse how this is done, also across the different organs, obviously. So if you cut something out of a lung, it's deflating, it's very different. If you have a beating heart, also again, very different. Here you see a kidney and it keeps its shape, but not perfectly so. So there are issues there which we are in the process of resolving. Happy to talk more about that. What we do have provided to the TMCs, and we ask them to use, is an interface where you identify the size, so the width, the height, and the depth of your tissue cube, your tissue block. It is here just 10 by 10 by 10 millimeter. And then you move with your mouse pointer that tissue block in a certain area, and then rotate it as needed, and then save out that location in a JSON data file. And we have for the first data release, you will have data for 24 kidneys and 24 spleen blocks that have been registered. And you see also here the position of these tissue blocks inside of those reference organs. And as you see, there are blocks registered for the top part of the left and the right kidney, and for also the lower part. And it's actually a question still, if these blocks up here are similar in their molecular and also cell type composition to the blocks in the lower part and left and right, etc. It's also still an open question. So now that that data becomes available, it will be very interesting to just look at this. We're also very interested in identifying spatial gradients. And so again, we are very much in the need to identify where these tissue blocks are that have been made available for Hubmap. Inside of the upload portal, the data ingest portal in Hubmap, you typically upload your tissue block data, but now you also have a chance to identify how many tissue blocks you extracted from one organ. And then you get as many registration opportunities, and you register the location for each one of those blocks that come from the same donor and the same organ. The new and improved version of the registration user interface, which will be coming available for Christmas this year, so in the second portal release. So some of you might think we are incredibly busy with the first portal release, and that's correct, but many of us are also working on our year 3 milestones. And so for the second Hubmap portal release, we will have an interface where you can turn on and off tissue blocks you have already registered previously. You will be able to also turn on and off different organ structures. So if you have certain markers that you are using in the interior of the kidney, you can see them by just taking away the outer shell, if you wish, of the kidney, and then registering your tissue block in there. You also see that certain tags are generated, not necessarily Taco Tuesday, but many others. So this is a mock-up of the final interface. And as you see, we are still trying to have some fun. You will also have an opportunity for non-Hubmap users to register their tissue blocks and to get that JSON file back. I don't think Homer Simpson is going to come, but we hope many of you come and register. And as you see, it will be supporting registration not just for the kidney, but also for colon, for heart, and for spleen. And actually those 3 organs come with predefined extraction sites, and you will be able to identify them. And in fact, there are different teams which have identified different heart extraction sites, and we will try to support multiple sets of its extraction sites. So if you're interested in learning more, please contact me. Going over to the exploration of this data, this is an earlier mock-up of the interface, or actually it's a screenshot of the current interface. And we are still hoping that some of the tiling will happen on the IEC side, in which case then thumbnail images will appear here, but we will also be able to zoom into these high resolution images that are generated by the Vanderbilt team. On the left-hand side, you see the portonomy for kidney being expanded, but also spleen, colon, and other organs are already in here. And these white blocks are the blocks for the kidney, which got registered, and you can click on any of those and retrieve data that exists for these tissue blocks. On the portal, on the Hubmap portal, you get currently to the CCF via this button. Again, in the future, we hope that it will be fully integrated into the portal itself, so that you can then use the semantic and spatial search within the portal functionality. You also saw that Ziv showed the sample data that Rahul Sahid and his team created at the other mapping component, which is very core and central to this effort, even though they're not presenting here today. What they are doing is to harmonize data across different data technologies, different individuals, different labs, which also might have slightly different protocols. Here you see 5 out of the 6 tissue data sets for which we have 6 single-cell RNA-seq data sets. You can then understand or use the spatial registration data to see where these tissue blocks came from. We also have metadata in terms of age and sex, for instance. And ultimately, the hypothesis is that those blocks which are more closely together, they would have more similar profiles in terms of their cell types. But also the tissue blocks from same age, same sex would have more similar profiles. And ultimately, you can also look at the cell counts and prediction scores across these 5 and soon 6 blocks. And actually, this can then be compared with the AST tables I introduced in the very beginning, where you can now see, all right, B lymphocytes, how does this connect to different anatomical structures, but also connect to the different biomarkers that exist or do not yet exist in Hubmap. So not all of the biomarkers potentially are run in every analysis. And as new types become available, there will be additional rows in the AST tables. Last but not least, I wanted to make sure that you saw that there will be a massively open online course that comes with Hubmap. We have been very proud to work with some of the major leading experts on this project to create a first set of 6 videos and modules, learning modules, if you wish. One is just an overview of Hubmap goals and ambitions. The other next module is a view behind the scenes at Vanderbilt University, how they create this really stunning data. Rahul is presenting data harmonization via Surat. And there's also a section on ontology design, 3D reference objects and user interfaces. There is, of course, a section on portal design and usage. And from what I understand, we will record this the day before August 4th, which is a weekend, from what I understand. I think there are many people very, very actively working on the portal right now. And there is also one more module on open consent your data, because if that data is open consented, we all can freely use it. And I think that's really in the spirit of open science and open data and open code and open education. So thank you so very much for your attention. We also, just like Niels, have a postdoc position available. So if you got interested in the AST tables and what to do with them, please contact me. I'm happy to tell you more. Thank you.
Thanks, Katy. So I think we have time for one question. So I'm going to choose. And so the question is, do you think that it's possible to automate the generation of these different ontologies? I presume the questioner means automate some of the different things that you showed, mappings and namings and things. If I'm wrong, Hiba, please clarify.
So what I mentioned was that we now have experts create these AST tables, and then there's data that also creates data-derived clusterings of cell types, for instance. So we could then use those data-derived cell type cluster hierarchies to the hierarchies in the cell ontology, and it will not be matching. But I think what we can do, thanks to having two visualization experts on the team, is to provide visual interfaces for experts to truly understand where the mismatches are and why they are there. And I think also we will need a ton of visualization to communicate data quality, confidence in certain types of protocols and certain types of workflows and provenance. We will need many visualizations to help people understand why certain views of our world might be valuable for anatomists, but maybe not for pathologists, and maybe not for those which want to plan surgery. And by the way, do not use these 3D reference organs to plan surgery. We have a big disclaimer, don't do this. So ultimately, I think there will be different views that are valuable for different biomedical research questions, but also for different means of improving health. And we will start with one reference body, just like we are currently using the visible human data to create a first draft reference organ set. But at Vanderbilt University, there's a sub-project going on to use CT scan data to create more consensus organs, which are relevant and valid for now 500 human bodies. And then as we go along and have semantic annotations and spatial annotations for those reference organs, which are consensus organs, and have more detailed AST tables and have the data-driven results, I think then we will really get to see where the discrepancies are. And I think it has to be not me, I'm more on the IT data science and engineering side. It has to be organ experts which have to ultimately agree how to name these things and what structures are most useful for what kind of question.